Thank you very much for attending and for your question yesterday. We're just going to finish the first part on the kinematics of locomotion and the role of sensory feedback. With me taking maybe 5, max 10 minutes of your time to finish this part and show how optogenetics can, based on informed and good analysis of the behavior, resolve the circuitry. So I'm just going to recap for everyone what we showed yesterday. So what we discussed, and here I put only the result for the silencing of the mechanosensory feedback that is excitatory, but it's very similar for the inhibitory one. What we discussed is the fact that when we're looking at animals in which the sensory feedback is silenced, and each point again on those graphs is one animal for which we do many trials of escape response, and we compare the siblings that are completely intact and the one where we use this botulinum toxin to silence just the sensory feedback during their locomotion. What we see is that we slow down the movement so that the speed is reduced during an escape response, and it's also associated with a reduction in the tail bit frequency. And if we go in finer detail during this bout, the escape response bout, what we see is that we have, compared to the control situation, without the feedback, we have fewer oscillation occurring in the fast regime that recruit the fast module of locomotion, including fast motor neurons and the corresponding premotor interneurons. And as a compensation, we have more oscillation in the slow component, so that the total number of oscillation, which is given by the hindbrain that commands the locomotion, is the same. It's typically 6, but what differs is that we're going to have fewer oscillation occurring above 38 Hertz and more occurring below, which lead to a reduction of the speed, and we have same distance travel but reduction, an increase of the duration of the total escape. And so based on this observation, we thought, let's look at the circuit that could mediate that. And we know now quite well that across all vertebrate species, they are in fact a very conserved class of interneurons that I introduced you to yesterday, that are premotor interneurons that will bring excitation to the motor neurons in order to drive them. So the motor neurons in humans, in mice, in fish, they're organized with this Heinemann principle that the large motor neurons are recruited for large amplitude of movements occurring at high speed. And the small motor neurons, the one that you see here, they are organized in a topographic manner in zebrafish, are ventral, and they are recruited for the small amplitude movement, which correspond as well to the slow motion, which recruits a slow muscle fiber. And so this is known for, indeed, like since the 60s in motor neurons. What was discovered by many groups I listed below is the fact that at the premotor level, the interneurons that drive the activity of motor neuron in the spinal cord, they are also organized with the same topography, with the large premotor interneurons being driving the fast modules, the fast swimming, projecting on the large motor neuron that are primary. And in contrast, the small excitatory premotor being ventral and being recruiting for the slow swimming and projecting on those guys, the slow, the secondary motor neuron. So we know very well that this organization is there. And so based on the effect we've seen, we postulated that the feedback should impact, if it was projecting directly onto the promoter pool, it should impact these guys, the dorsal like V2A interneuron involved in the fast swimming, because we have a change in tail bit frequency. If they were just projecting onto the motor neuron, we would expect a change in power of movement, the amplitude of movement, without changing the frequency itself. The fact that we had a very specific effect on this fast swimming frequency made us think that it should be projecting on those guys. And so we went, and thanks to the power of the genetics in this transparent animal model, the larval zebrafish, we have a chance to actually really take a look at that. And the anatomy is often, after the behavior, the second main source of the answer for the mechanistic understanding. So we saw that indeed those sensory cells in green here, they have axons running right next to those large dorsal motor neuron, dorsal premotor interneuron that are excitatory and recruited only during the fast swimming. And so their axon here is compatible with the position of those dorsalmost V2A neurons. And therefore, we took a look using optogenetics by using an opsin, CoChar in this case, in the sensory cell. And then we check whether we see that this sensory neuron is indeed projecting onto the dorsalmost V2A interneuron, and whether it can recruit it, elicit spiking quite rapidly. So we activate, with optogenetics, the sensory neuron. We show that we have a very, nice signature of this sensory cell. We can elicit single action potential. And then when we've done the experiments of activating the premotor cell, the sensory cell, and recording from the premotor interneuron, we see that in those V2A interneuron dorsalmost, upon excitation of the sensory cell, we get a very clear EPSC, so an excitatory postsynaptic current that is elicited right after the pulse of light, meaning that it's monosynaptic. And what is remarkable is that it's very steady, so that every stimulation that we repeat, we're getting a clear response. If we block the excitatory synapse, we don't get any more this response. So it's mediated by a glutamatergic synapse. And what is interesting is that even though this neuron here is very large and typically difficult to activate, the connection from the sensory cell onto the premotor interneuron is very effective. So that one single action potential from the sensory cell is sufficient to elicit a current that will drive the action potential in the premotor interneuron, those V2A cells. So thanks to this approach, guided really by the behavior and the fact that we looked at the fine kinematics in order to understand the mechanistic, we've been able to reveal a circuit by which the sensory cell project directly onto the fast module of premotor interneuron that themselves project widely onto the motor neuron pool in order to get the fast swimming. And so here are the people that have done the work. In particular, I talked about the work of Stephen Knafo here and Kevin Fiedlin, who were the people who really focused the most on this sensory feedback, together with Urs Bohm, Lydia Genoun, Jeff Hubert, Jonas Stenberg, who were pioneers in investigating this new sensory pathway we have identified in the vertebrate spinal cord. And then if you're interested, we're actually hiring, so please share the words. We're looking for an engineer in optics and hardware and a postdoc in electrophysiology, so feel free to contact me. And now I will leave the floor to the tales of the two Gautams, and we'll start with Gautam Reddy. I introduced you, before I started a little bit. So Gautam Reddy and I have met during the school of the Kavli Institute of Theoretical Physics in Santa Barbara, where I was coming with zebrafish to do experiments on pH taxis, and Gautam was one of the 4 students who joined the group in order to understand, well, how can we understand the long-term dynamics of animals when they're not looking, we're not just looking at single bout anymore, but we're looking at sequences of bout. And this is, in fact, often the case for many of the response that we're looking at. So the method that Gautam Reddy has developed is working well for zebrafish, but hopefully you will see that it can be very much of use for other animals. And we're really excited to try it on other animals. You will maybe mention that also. And then Gautam Reddy was attending the school between his PhD and his postdoc, but he's now a postdoc, I believe, at NSF fellow in Harvard University. So he can maybe answer also question if you have any about that. And he will be followed by Gautam Sridhar, who is a grad student in the lab from our program called Zenith for Interdisciplinary Neuroscience. And Gautam Sridhar basically follow up on the steps of Gautam Reddy, applying the same segmentation methods to new kind of behavior, including precapture. So we will show you that. Thank you very much for your attention. I will stop sharing. And maybe now, Gautam, you can start sharing.
Hi, everyone. Just to give some background, my background is in physics, actually. And but during my PhD, I was working on different problems in animal behavior. So when I say animal behavior, it's not really coming more from the neuroscience side. I've been looking at animal behavior purely from the point of behavior itself. So thinking about what we can do just by observing animals. And essentially, this field is a rapidly developing field. I think there's plenty to do, especially for physicists and computer scientists. And it's really, I think, driven by the fact that we can do things like you see in this video here, where you can track animals very easily in the wild, capture their postures, and analyze high dimensional data. So in this lecture / tutorial, I think I'll spend half the time talking about animal behavior in general, a little bit of the history of the field, what's happening right now, like why there's so much activity in the field right now, some examples where modern methods have been applied, and some interesting insights have been gained in animal behavior. And the second half will basically be about the work we did with Claire in zebrafish. So animal behavior is of course not a new field, right? Like it's been going on for a long time. People have been observing animals, describing what animal behavior, how animals behave when you give them certain stimuli, and so on. So these three folks are some of the pioneers of the field of ethology, right? The study of animal behavior. They received the Nobel Prize in 1973. And Tinbergen especially wrote this fantastic book called The Study of Instinct, which is a classic in ethology. And in this book, Tinbergen highlighted 4 questions that could possibly guide researchers in ethology. So Tinbergen essentially said that there are 4 kinds of questions you can ask in ethology. And I think these questions actually apply more generally in biology as well. The first one is function. You have causation, evolution, and development. And the way I like to think about these questions is to think that when you're trying to describe the behavior of an animal, right? So the animal receives some stimulus from its environment. It executes some response in response to that stimulus, and for that response, it gets a reward, right? So the first of these questions, that of function, is really to ask, how is the reward shaping the stimulus response function, right? So the question is it's a normative question of how the fitness is related to the response of the behavior of the animal. The second question is that of causation, which is to ask what stimuli evoke what responses, right? So it's just mapping out the relationship between stimulus and response. Third one is that of evolution. So how does the stimulus response evolve in the first place? And the way we usually study this, of course, is to look at the same stimulus response function in closely related species, for example, or different kinds of genetic variants as well. The fourth question is that of development. How does behavior change across developmental time within the time scales of a single organism? Right? So these are the kind of questions ethology has been asking for many years. Most of these questions for many decades have been asked simply by looking at specific animals over the lifetime of the researcher, essentially, right? So because these are difficult experiments, you need to manually observe these animals, keep track of what they're doing, apply specific perturbations. But this has completely changed in basically the last 15 years, and I'll try to explain why. So here comes the field of modern computational ethology. And I really believe this has been enabled by various very recent advances. The first one is the fact, of course, that we can record videos, right, in high resolution, both in time and in space. And I don't really need to explain that. The second one is the fact that we can do posture tracking automatically, just like the video that you saw in the very first slide. The third one is the fact that you can take this posture data, which is presumably these are what the output of this posture tracking are points in some high dimensional space, right? So you can imagine that as the animal is moving, you're tracking the dynamics of the animal in some high dimensional space. And now there are methods to actually analyze this kind of high dimensional data, right? Using methods from machine learning. And given postural data analysis, you can then ask, is there some kind of structure in this data? Can you identify patterns in this data? This pipeline is highlighted in this nice review here recently. This kind of highlights the various steps that one would go when you typically try to do a purely behavioral study, right? And there are also certain methods highlighted here which tackle various parts of the pipeline, right? So the first aspect is just to record the animals, right? So video recordings. And then you extract certain features, right? So if in this case, if you're interested in the mice, you would extract, let's say, the boundaries of the mice. But of course, these features are usually high dimensional. So for instance, if you look at this, I guess there are around 7-8 points for each mouse. And so these are embedded in some high dimensions, and you'd like to reduce the dimensionality of these feature space so that you can visualize the temporal dynamics. Right, so from there, you get a low dimensional representation of the temporal dynamics, like the kind that you see here. And then you could do various things. You could do certain modeling. You can do clustering. For instance, if you do clustering, you can identify potentially different kinds of behaviors. In this case, a mouse sniffing or a mouse turning and so on. And there are many algorithms. I think this is really a small subset of the kind of methods that are coming out right now. There are like 4 or 5 methods coming out every year, including ours. And they differ in what stage of this process they tackle. There are some which use methods that go straight from the videos directly to the trajectory analysis, like Murthy from Bob Detta's group. But each of them might tackle different stages of this pipeline. Just to give you a good example, what I think is a good example of this pipeline working from start to end. I'd like to show you this example from trail tracking in carpenter ants. This is an experiment from Venkatesh Murthy's lab in Harvard, which I collaborate with quite often. You might have met Venki at the very first day of the lectures, I believe. Right. So this is a carpenter ant. It's literally found near the river in Cambridge at Harvard. This is taken in the dark. It's under infrared lighting. Ants don't see in infrared, so they're in the dark, and they're tracking a pheromone trail, right? So this is camera from the top, and the stage is moving. Maybe I'll play that again. Right, so it's tapping its antenna in order to track the trail. Right, so I mean, trail tracking is a very common phenomenon. I mean, we've pretty much all seen ants track trails, but it turns out that we don't really understand how exactly they're following the trail in terms of navigational algorithms. And this paper set out to kind of understand that. And the idea here is that you take images of this sort, right? So you get a time series of this sort. You can use certain computer vision based methods using neural networks. And the basic principle behind those methods is that if you can train the neural network to identify certain body parts. So let's say you can train the network to identify antenna. So specifically that would mean you would put markers on the legs or the antenna for maybe a few hundred frames in this video, or usually longer videos. And then the neural network essentially learns to figure out where the antenna and the legs are, or the thorax is for different frames, right? So completely different frames. So it can generalize for new situations. So this method, I'm showing you this method just for the tips of the two antenna, the head and the body of the ant tracking the trail. This is the kind of output that you would get. And as you can see, we're really just limited by the temporal resolution of the camera. Otherwise, we can track this really well. And there are pretty much no errors. Kind of see the ant touching the trail and following it. It's a curved trail. You can also track the speed of the ant, and you can see these patterns of it slowing down and going up and down. And the fascinating thing about this, I think, is the fact that you can really look at the microscopic aspects of the behavior, as well as the global aspects of the behavior. So if you actually look at this trajectory and plot it out in a single figure, in red is the body, and the two colors are the antenna, and the black is the trail. You can see that the ant is actually pretty much on the trail, right? Like it's in the dark, so it cannot see the trail, but it's able to track the trail. And if you look at the trajectory as a whole, which you really only do by tracking the antenna continuously, you see that there's in fact a stereotype pattern that the ant is doing with its antenna. It does like a left motion onto the trail with the left antenna, and then it does the right, and then it does left, and then it does touches with the right, touches with the left, and so on. It's doing what I like to call like a windshield wiper motion in order to keep track of the trail. You can do this. You can do a little bit deeper analysis into this. So, for instance, it turns out that there are different kinds of trail tracking behaviors that the ant does on different kinds of trails. This is a straight trail, and just to illustrate the different kinds of behaviors that it does, it's an example where it showed like the 3 different kinds that we observed in experiments. So it's one of the behaviors is a sinusoidal motion where it tends to move its entire body around the trail. As you can see here, if you look at the distance from the head to the trail, it's like a sinusoid. At some points, it turns out that it just stops, and then it taps on the trail. It almost feels like it's trying to figure out how the trail feels like. It spends quite a bit of time doing that. This is this black area here. And then this blue area is the trail following motion that I showed you, the windshield wiper. And the general idea is that once you get these trajectories, you can apply some clustering algorithm, and you can identify the various patterns in this behavior. Specifically, in this case, these are the 3 different kinds of trail following strategies that I just showed you, sampling strategies that I just showed you. So the probing one, where it's tapping close to the trail following, where it's doing the windshield wiper, and the sinusoidal, where it's expanding its antenna and then it's going around the trail. By the way, feel free to ask questions, interrupt me. I should have mentioned this before. Okay, so the idea that you see characteristic patterns in the data, like in the ant's case, is not new at all. People have observed this in many different animals, and I think it's quite intuitive from our observations of animals as well, right? This is a classic example of stickleback mating. This is the model organism studied by Tinbergen, who I mentioned in the first few slides. So Tinbergen, in this case, was describing this complex mating ritual of male sticklebacks. So they would build a specific kind of nest. They would change their color, ward off any other competitors, then do a specific dance to woo the female, who accepts by making the specific motion. And then the male would guide the female into the nest, prod the female, which would lay eggs, and then the male would take care of the eggs, right? So in this case, it's kind of circulating the air to make sure that there's oxygen supply. And the important point here is that this is a very stereotypical pattern, right? So if you take a stickleback, put it in an aquarium with some sand and so on, it will do the same exact behavior. If you use like a red object, male sticklebacks are red in color. So if you get a red object near another male stickleback, it will show some aggressive behavior. Or you could prod the female with a rod and it will still lay eggs and so on. So essentially, you can think of this behavior as a sequence of programmed actions, right? So it's a programmed action sequence, and there are plenty of examples of this. This is another example that I'm familiar with from my PhD thesis, which I worked on soaring birds, right? So if you look at soaring birds, which I'm sure all of you have seen, they climb thermals in the atmosphere to gain height and migrate. Here you're seeing a soaring falcon, the GPS trajectory of a soaring falcon. And you can see these spirals of it going up, and you see them all over the place. It's a characteristic pattern. And when you see a bird, we've all seen these spirals. But the question is whether we can infer these action sequences from raw kinematic data, right? So given that we can simply observe animals, track them, how do we identify these sequences without actually looking at the trajectories themselves? That's the question that we would like to answer. In order to tackle this question, I collaborated with Claire, as Claire was mentioning. This is in larval zebrafish, of course, because that's what Claire's lab does. This is a rather simplified setting, but I think it illustrates the basic difficulties and the challenges of inferring action sequences. This is a case where a fish is in this rectangular tank here. It's about 15 centimeters in length. There's acid on the two ends here. Fish don't like acid, so they will try to escape it. And fish swim in bouts, as Claire has probably mentioned. So the fish is moving around here. It gets close to the acid, and then it performs this escape sequence here. So the fact is that fish seem to be performing some sequence. You can look at many videos, and they seem to be performing a sort of a similar sequence across different fish and within the same fish. If you look at how much time they're spending within this arena, in the case of exploration where there's no acid, you see them spending pretty much the same time everywhere. But if you put acid, they're avoiding the ends. So they're doing something to avoid the ends. But if you look at certain global kinematic parameters, for instance, if you look at the average speed or the distribution of speed of the fish, the change in heading per bout or the tail attributes of the fish in these two environments, with acid and without acid, you can't really tell the difference. Right. So the point is that if you look at global parameters, you can't really tell what the fish is actually doing. So the question then is, how do you infer the specific sequences that the animal is doing the fish is doing in order to solve the task? So it's really a problem of inference. In zebrafish, things turn out to be a little bit easier because there's a clear analogy between a zebrafish behavior and language. And Claire must have explained this to you before. And that's because zebrafish swim in bouts, and these bouts are, for the most part, stereotypical. Right? So if you track zebrafish and you look at the tail segments, if you track the different tail segments here, and if you plot the tail angle over time, you can imagine this as the angle between the body axis and the end point of the tail. So this is the theta that I'm plotting here. They tend to beat their tail. Sorry, I hear a question. Go ahead.
Yeah, hi. If you go back to the previous slide, in the case when there is no acid, have you looked at things like mean square displacements and compared them with the case with the acid? Do they also show similar trends you can't make out?
Yeah, so we looked at a bunch of variables, and most of them look like this. And mean square displacement, for instance, there are minor differences, as you can see here. It goes slightly faster. So the differences are there, but they're minor. So we know, for instance, that they're executing more fast bouts and slow bouts, and they're moving longer during each bout, but we don't know exactly where they're performing those bouts. And what sequences are being performed to escape the acid.
Okay, but even in the case when we don't have any acid, is the motion diffusive? I guess not.
Yeah, we haven't really looked at the statistics of the sequential statistics, whether it's diffusive or not. but I would actually say it's most likely not diffusive, because they tend to do a lot of the same forward bouts repeatedly. So they're
Right.
not changing their direction. They have a tendency to repeat the same bout.
Right. Thanks.
Okay, so if you look at the tail beat over time, you can see that they swim for 100 or 200 milliseconds, and then they don't swim for a few hundred milliseconds. So there's a rest period, and then swim and they swim, and so on and so forth, right? And if you look at each individual bout and you kind of zoom in, these need to be taken with a high resolution camera because the tail beat is really fast. You can apply certain clustering algorithms to figure out if there are certain characteristic bout types. In this paper, they developed their own clustering algorithm, and they had fish behaving in many different conditions in order to characterize their behavior as a whole. It turns out that they can identify, I think 14 of these bout types that you see here. Right, that's 14. And some of these are pretty similar, right? So if you look at some of these bout types, they look quite similar. For instance, the J turns, high angle turns, and so on. But the point is that there's a higher likelihood of seeing certain bout types. For instance, these are capture swims that fish use when they're executing prey capture. That fish would execute in certain contexts more often than others. The point here is that if you look at the sequence of bouts and spacings, you can really represent it as a sequence of characters, let's say from an alphabet, which is why I mentioned the analogy with language, right? So you essentially have here like a slow, a burst swim, and a different kind of bout, and slow, and so on. So there are different ways of describing transitions within this behavior, and the typical way is to plot what is known as an ethogram. And I think in classical studies, what an ethogram meant was basically like the fish builds a nest, and then it woos the female, and if the female so it's basically a decision tree that is explaining the program, the behavioral program that the animal has. I think in modern terms, how people think about it is basically a model of the dynamics, right? So in this case, it's specifying the probability of going from one bout to another, and this ethogram is going to differ in different environments, and you can possibly compare across environments and so on. In this case, this is equivalent to a Markov model, if that is helpful. But what we'd like to argue, and we looked at this kind of ethograms. When I say we, I mean Claire and I. We looked at these kind of ethograms, and we realized that these kind of ethograms really don't give you the kind of information that we're looking for. These don't really tell you whether there are abnormal sequences or rare sequences that are solving the task, right? In this case, the pH taxis task. And in order to kind of illustrate the issues with these kind of models, imagine that you are given a behavior of simply someone tossing a coin 100,000 times, right? So you obtain a large sequence of heads and tails, but in fact, within those 100,000 sequence of heads and tails, there's actually a sequence of many tails consecutively, which is quite unusual, right? So this sequence is unusual, but since it's embedded within this long behavioral data set, you can't really identify it. And if you apply these kind of models, they will show you that tail to tail is slightly more probable than head to head or tail to head and so on. But it doesn't tell you that there's actually this weird sequence of 20 tails embedded within this sequence. So we wanted to develop methods that can identify such sequences. So just to illustrate highlight the dichotomy of using Markov models versus identifying sequences, I'd just like to show you the classic model that people typically use to analyze behavior. Hidden Markov models have been developed, maybe they've been reinvented many times. maybe about 40, 50 years ago in the context of speech recognition. Sorry, I have a question.
Yeah, sorry, just to clarify, with respect to the analogy to the coin toss, but the assumption here is that the individual, so in the coin toss, each coin toss is independent, whereas in the behavioral ethogram, we're actually doing away with that assumption. Is that right or not?
Yes, in the coin toss, they need not necessarily be independent. I was using the independence as a just a simple example, but I'll show you that there's an independence assumption that we assume in our model that helps us solve the model, but it's the dynamics itself is not independent. So we're not going to assume that the bouts are drawn independently.
Okay, thank you.
Still a dynamical model. Right, so hidden marker models have been there for a long time. And the basic idea of hidden marker models and many kinds of inference based models is that you observe, sorry, another question. The basic idea is that you observe a behavioral output, and this is not necessarily a sequence of letters from an alphabet, but it could be some high dimensional sequence of behavior, or it could be speech in which it was first developed. And the idea of these hidden models, or when they say hidden or latent models, is that there's some kind of hidden process that is generating this the underlying, the observed behavior, right? Which you cannot observe. In this case, it's the mapping from letters to behavior. In this case, it would be bout types to actual bouts that you see, and the transitions between bout types. So the goal then would be to write down this model. Given this observed behavior, you'd like to infer the structure. My headphones automatically connect to them. Sorry about that. All right. Yeah, so the idea is to infer the characteristics of this model given the observed behavior. In BASE, which is the algorithm that we developed, we call it behavioral action sequence segmentation. The idea of BASE is quite different from hidden marker models. In BASE, what we do is that we don't assume that there's some kind of transition dynamics going from one bout type to another. So in this case, we are trying to describe a transition dynamics. What we instead do is to assume that behavior is drawn from some motive templates, right? So there are certain sequences of behavior that the animal is executing, and then it's executing these sequences one after another independently. So that's where the independence assumption is, right? So you can think of these as words from a dictionary. So AABC would be a particular word, AB would be a specific word, and then you're drawing these words sequentially from this dictionary. And each word in its particular instantiation of the word, like in a particular example or realization, it could be not exactly that word. You could kind of misspell it, or you can not execute one of the characters. So there could be some noise. But given that instantiation, you would get some observed behavior, right? So there's again a mapping from A to YA, B to YB, and so on, which is essentially the same as hidden marker models. But the key assumption is that instead of the transitions between characters, we're really describing behavior as a sequence of words from a dictionary, right? And the goal again is to invert this process, right? So given this observed behavioral output, you would like to go back and identify these two arrows here. So in the interest of time, I'm just going to give an example of this algorithm applied to English text, and Gautam Sridhar is going to explain applications to zebrafish behavior. And the example I'm showing you here is purely in English. But you have to imagine that each letter is associated to a certain specific behavior. So in this case, I've taken this fraction of Pride and Prejudice, right? removed all punctuation, removed all spaces, no capitalizations, and then you get a sequence of letters like this. And given this corpus of characters, you would like to identify words from English. You'd like to learn language, given that you know the alphabet, right? So you know A, B, C, D to Z, but you don't know the words. So you need to figure what the words in language are. This algorithm has been developed a while ago for bioinformatics, and we are really inspired by this algorithm called Moby Dick, which was developed for essentially to analyze DNA sequence. So how would one go about doing this, right? So one way is to just look at these different characters. If you didn't know that these were actual words, the best thing you could do is for look for patterns, right? So you try to see if there are certain combinations of characters that are occurring more often than you would expect, right? So initially, like let's say A happens with probability 0.5, B happens with probability 0.2, but AB is happening with probability 0.9. So somehow AB is occurring a lot more often than A and B are happening individually. Then you would probably think that AB is a word by itself. That's the basic idea. And then you add this word into the dictionary. So let's take a simple example where you only have 2 characters in the dictionary, in the alphabet, sorry. So let's say you observe the sequence. This is the data set that you get. So the idea of Bayes is to start off with an initial dictionary. Usually you start off with a dictionary with only characters. So let's say A and B. You wouldn't put AB from the very beginning. If you have only A and B, you would first ask, what is the probability of A and what is the probability of B? Which means that what is the probability of drawing A from this dictionary at each instance? So if there are only A and B, you would simply count the number of A's, the number of B's, and divide by the total number of A's and B's. So in order to get the probability of A and probability of B. If you have A, B, you could do the same. In order to calculate these probabilities, we use a maximum likelihood inference method. I think, again, in the interest of time, I can't really go into how exactly we do that. But the basic idea is to calculate these probabilities. These are not frequencies, by the way. Like frequencies are different. Frequencies are simply measuring the number of times AB appears. We don't want to calculate frequencies. We want to calculate probabilities of the generative model, which is to say, if you partition this data into words drawn from this dictionary, in how many partitions would you expect to see AB how many times, right? It's a subtle difference, but it's important because that's how the generative model is framed. And it turns out that computing all possible partitions of the data set is difficult, but there are efficient ways of doing it. And the details are in the paper. So the idea is that given that you have some probability of A, probability of B, and AB, you would then add new words if you think that certain combinations of A, B, and AB are occurring more often than you would expect by all possible ways you can form A, B, B, right? So in this case, if the number of times you see A, B, B is much larger compared to the probability of seeing A and then B and B, or A and then AB and then B, right? So these are 2 ways you can form A, B, B. If this is much greater, then you would add A, B, B into the dictionary. Once you add A, B, B into the dictionary, you recalculate the probabilities and throw away the words that have extremely low probability, that they almost have 0 probability. You put some threshold and then throw them away. And then you repeat this process. I'd like to highlight that this kind of, yeah.
Sorry, there is a question. Arnab is asking, are the partitions equal or variable in length?
Partitions are variable in length. So in fact, there are an exponential number of partitions. If the sequence is of length L, there are 2 raised to L minus 1. You can calculate it. There are 2 raised to L minus 1 partitions, and they can be of different lengths.
Thanks.
All right, thank you. And then you cycle between these 3 iterations, and eventually you will converge on a dictionary. I like to stress that this idea of starting off with a specific model, inferring the best parameters, and then improving the model in a different dimension, in this case, adding new words in the dictionary, is a very standard method. It's formalized in certain inference methods using something known as the EM algorithm or the expectation maximization algorithm. And it can be given a proper theoretical framework. And a lot of the methods in machine learning, which have this kind of inferential structure, you can make this arbitrarily complex, by the way. Nowadays, you can do really complicated models going from latent dynamics to observed behavioral output. And it really depends on the organism that you're looking at, I think. But most of these algorithms rely on this kind of idea of maximizing some parameters of a generative model and then refining the model and iterating over this process. I actually had an example that I was going to show you on live on a small data set, but I think I'm going to skip that because there's probably not enough time right now. But I'll show you the results that come out of applying this algorithm on this text. But really, like, you have to imagine that each character is actually a behavior. So we're not actually seeing this kind of data set. We are often seeing like a high dimensional sequence of vectors, right? So it's some dynamics in high dimensions. Okay, so this is what you get when you apply this to the first 10% of Pride and Prejudice. These are the top, I think, 25 or 30 words, and they're scored by how unlikely they are if you randomly concatenate different characters. As you can see, I mean, this is biased towards longer words because these are more likely to be unlikely if you randomly concatenate stuff. But you can see that it's basically capturing a lot of the proper nouns, which have unusual combinations, right? Like R, S, B is probably highly uncommon. But these words are actually not appearing too often either. So in many cases, we can show in other examples as well that this method is able to capture unusual sequences, even though they occur only a few times in the data set. I think Gautam Sridhar will also show some of that data in Zebrafish. Yeah, I think that's it for my part of the presentation. I can take any questions now, or I can also do it throughout. Yeah.
Maybe it's good to take a few questions now before we switch to Gautam. Sridhar?
Okay, any questions? Maybe I'll start off. So taking this analogy to language a bit further, so as you nicely put in this list of words from literature, so the concatenation of words follows a certain grammatical order, right? So how do we translate that into behavior where we say that, you know, certain syllables or certain words are concatenated with much more frequency because that's what makes sense for the behavior of the animal?
Yeah, I think so. The specific model that we base specifically does not do that. And you can, I think maybe there's an example here. Yeah, so for example here, the country, right? So it has concatenated two words that are really distinct words. And the reason is because the country happens very often. But you could, in principle, add dynamics between words. So you can add some syntax. So this one has no syntax whatsoever. It's really, I would say, like a lexical. We call it a lexical model because there's purely lexical model because there's really no syntax. But you could in principle write down an arrow here, so the specified dynamics between different words. I think that I haven't tried this yet, but I think the model becomes a bit harder to calculate because now you have n square parameters where n is the number of words in the dictionary. And when you're looking at a limited behavioral data set, it becomes hard to infer both the words, the probability of the words, as well as these n square parameters that you have, extra n square parameters. I think you can do it given a large enough data set for sure. The question is whether within the limitations of the data set that we had, we could do it. But I think in theory, it's possible.
What about the converse? That is, if you have a certain behavior or action sequence, that if action sequence A occurs, B will definitely not occur at this time point. So going back to the Auger paper, for example, between escapes and routine swims and routine turns and so on, you can sort of say that when the animal is trying to escape, it probably does not do J turns, right? So ruling out the words that are not likely to occur at all, sort of like suppressing syllables if one syllable occurs.
Yeah, that's a great point. I haven't thought about that in that specific way. I guess it's in some ways it is a combination of. So I guess one way to think about those, this model does not capture that. Just to be upfront, I think the one way to capture that would have another layer here, where in each motif you only draw a certain subset of bout types. So let's say there are like two classes of bout type of words, and in one class you only have slow bout types, and another class you only have fast ones. And then there's another layer that you could possibly infer. And given that class, you only spit out a word that is has bout types of that class. I can imagine doing that. I think that's a great point. Yeah, it's like a clustering algorithm on words.
Thank you. Vijay has a right.
It was
Vijay has raised his hand. Yeah.
Yeah, so it's probably related to Vatsala's question. So I was trying to think, is the map between the microscopic template that you're showing here and the behavioral output, is that a deterministic map?
Template to output, no, each of these is a probabilistic map. So here you can have the transitions from template to instantiation, where you can have insertions or deletions, like I was showing you here. This is probabilistic, so you have some probability of insertion or probability of deletion. You can change those parameters in this model. And then the map from instantiation to output is also probabilistic. So it's a, in this case, as Gautam Sridhar will show you, it's a Gaussian mixture model. So A maps to some high dimensional bout type A, and that's given by a multi-dimensional Gaussian. I think he might explain in more detail